You have a prototype of your dream cosmetic product. You have your vision board set up, but you feel that you lack some understanding regarding formulation and lab testing. This preclinical stage is really where your product starts coming to life. It's a stage where you will need experts, research labs that are really specialized based on the claim that you want to support. I'm Emily, product development consultant at Cosmetic Lab. With a background in biology, genetic and biotechnology, and several years working as a business developer and project manager that help clients develop their products, I'm here to help. I see that you are serious and committed to develop your cosmetic products. Let's now talk about this really exciting stage, which is preclinical research. The heart of preclinical research is formulation. Formulation is both an art and science. It's about finding the right balance between safety, efficacy, stability, and sensory appeal. You will have to find and select the right ingredients that are in phase with your concept that you have developed during the discovery phase. Of course, in order to have the best formula, you will have to go through different prototypes because you don't know at this stage how the ingredients will interact with each other. But don't worry, you will not face this stage alone. Different organizations can help you with this stage because it really needs a specific knowledge and extreme accuracy to develop a formula. You may want to look at formulators or chemists or contract manufacturing organization. Even sometimes raw materials suppliers can have in-house the facilities to develop formulas. Check out cosmeticlabs.com with a directory of hundreds of research labs that can help you with this stage. Great, now you have your formulation. Finally, an outcome that you can hold in your hands. But it's only the beginning for your formula. You wouldn't want your formula to deteriorate just by sitting on a shelf, right? Or to give your future consumer rashes. So let's do some safety and stability tests. So regarding stability tests, your product will endure different stress conditions. Your product will be exposed to light, to cold, to heat. Your product also needs to be checked if it changes colors or texture over time, and you will have to determine the approximate shelf life of your product. Regarding safety, you can do some preliminary tests to test the potential risk of skin irritation or toxicity. You can also do some compatibility testing between your product and the packaging. Some microbial testing will also be needed to be performed in order to see if there is any bacterial growth that happens in your product over time. You may need to compile all this data into a regulatory file for each ingredient, especially if you are using components that are not commonly used in the cosmetic world. Okay, safety and stability is really important, but let's not forget that you are developing a cosmetic product that is supposed to solve some issue that you have included in your vision board during the discovery stage, right? For that, you will need to test your product on skin models that several research labs are able to produce in order to test the efficacy of your product. I should warn you about something. Depending on the composition of your product, there are two ways to go to this efficacy testing phase. Either you test directly the formula, either you test the active ingredient or the mix of active ingredients first. Why going a step back by testing the active ingredients, you may ask? Very valid question. Indeed, as for some of my previous clients, it's not very intuitive. Let me give you a case study. During the discovery phase, you have chosen an ingredient that will be the main or maybe even the only ingredient of your formula that is trending right now. Let's take, for example, the ascorbic acid or vitamin C. So your main claim is that your product should have antioxidant effects, right? Nothing really tricky, as you may know that vitamin C is the most powerful antioxidant. However, it happens that when formulated, the active ingredient that show great promises at first decrease in its potential and its value. In this case, you will see a significant decrease of the antioxidative activity of vitamin C. If you still have an effect, that's great. If your effect is close to zero, not that great. Okay, Emily, you have demonstrated that efficacy testing is really important. Not that you should test active ingredients. Yes. But if, for example, in another case, you have formulated vitamin C with other compounds, a compound X and a compound Y. Separately, all these ingredients show great results, a great efficacy regarding the claims that you have chosen. But once formulated, you see that vitamin C has zero effect, that the compound Y brings toxicity, and that the compound X has a huge decrease in its efficacy. Is it because the formulation interacted with the compounds? Is it because the formulation interacted with the ingredients? Is it because the compound X deteriorated the activity of the compound Y, and that the toxicity from Y decreased the antioxidative activity of the vitamin C? You see where I'm going? In my opinion, by testing the ingredients separately, then in mix, you can see if there will be any interaction between them. And then you can move to the formulation to see if the formulation also interacts in a negative way your different ingredients. It is how I would do it. So efficacy testing is really important. A research lab will offer you skin models depending on what you want to analyze, what claim you want to validate. For example, in vitro models such as cell culture, such as lab-developed skin models, or ex vivo, meaning on skin tissues or other biological samples that have been collected on human beings. And depending on what the cosmetic claims are for your product, you will be suggested different endpoints and readouts. To avoid any confusion, an endpoint refers to a specific measurement or outcome that is used to assess the efficacy or effect of a product. A few examples will be skin hydration level, or the improvement of skin elasticity, or change in the skin pigmentation. Readout refers to the actual data that are obtained from a measurement or observation in a study. For example, a readout for a hydration level will be the quantification of hyaluronic acid. A readout for the skin elasticity will be the quantification of the protein elastin. For the change in the skin pigmentation will be the expression of the gene coding for melanin. To find a research lab that can help you with all these skin models, because there is a lot of skin models, a lot of endpoints and readouts, you can check out cosmeticlabs.com. You will find a directory of research labs filtered by country, and soon to come, a web app where you will be able to find a lab based on the claim that you want to support, based on the models that you are interested in, and more. You will find a link of the waiting list. You will be informed when the app is ready to be launched. Remember during the discovery phase when we talk about the feasibility assessment and if your product could be manufactured at a large scale? Well, based on the result that you have obtained during this efficacy testing and safety and stability testing, you may want to refine your formula and rethink a few things. If the concentration of ingredients was already maxed up in the formulation and you don't see any results, well, it may be complicated to scale up the production of your formula if you need to increase the concentration of your ingredient by 50-fold. And why the clinical research is really important, because it sets the foundation for the clinical trials. Yes, because next step is testing your products on living and breathing human beings.